Hello everyone, welcome to this session, and we should be all in Phoenix, but unfortunately we are online. So today I'm going to talk about pharmacological strategies for therapies targeting PTC mutations. These are my disclosures. So in the case of cystic fibrosis, we have 7 functional classes of mutations which have evolved into theratypes, meaning that mutations within the same class or theratype will actually be treated by the same therapeutic strategy. So today I'm going to focus on class 1 mutations, and these are the ones that actually have a stop mutation. So examples include G542X, R1162X, or W1282X. So we expect that these mutations can actually be rescued by what we call PTC suppressors or read-through molecules. So mutations in this class actually lead to a premature stop codon or termination codon, in short PTC. So these mutations actually lead to the introduction of this PTC, this premature termination codon, so that when messenger RNA is being read by these protein machines, which we call ribosomes, they actually stop in a premature way, leading to the production of a truncated protein. However, the cell has a surveillance mechanism called nonsense-mediated decay, in short NMD, which actually prevents the production of these truncated proteins, which are potentially toxic to the cell. So that messenger RNAs, which actually have a premature termination codon or a PTC, undergo this mechanism of NMD. The goal here is actually to identify small molecules which mask these premature termination codons so that an amino acid is actually introduced in the polypeptidic chain, the messenger RNA degradation is avoided, and the full-length protein is actually produced. So examples include molecules like the PTC124, also known as ateleren, which was tested in the context of cystic fibrosis in the past, but was actually discontinued for lack of significance. So the model for the decay of messenger RNAs by nonsense-mediated decay is rather complex, but it includes a number of factors and actually depends on a complex which we call, marked here in blue, and called EJC for exon junction complex. This complex of proteins is actually deposited at exon junctions during the splicing process, and it is removed by the ongoing ribosomes, which actually translate messenger RNA. So this recognition of substrate messenger RNAs that are to undergo nonsense-mediated decay is actually through the presence of this exon junction complex, which is not removed by the ribosome because the ribosomes have dissociated before due to the presence of the PTC. So this slide is actually illustrating the reduction in the levels of messenger RNAs with PTC mutations versus mRNAs, also some CFTR, but without PTC mutations. And on the left of the graph, we're showing the differences observed between PTC and non-PTC in primary respiratory cells, both nasal and bronchial. And on the right of the graph, we observe the same difference occurring in messenger RNAs from organoids. Here we observe for primary nasal cells that the levels of messenger RNAs with PTCs, that is the middle bar, are significantly reduced versus the levels of messenger RNAs without any PTC mutations. So in short, we can conclude that messenger RNAs with PTC mutations are degraded down to about 30%. And this implies that besides read-through agents that rescue the PTC mutations, these mutations need to be rescued in terms of the degradation of the messenger RNA from NMD. So in order to identify genes that actually affect NMD and or read-through, we have used a mini gene construct. Actually, it's a little bit complex, but in short, it has an M cherry at the N terminus and an EGFP, so a green fluorescence tag at the C terminus. In the mini gene, the M cherry is on 5' and the GFP on the 3'. And the GFP is actually just before the final stop codon. So in order to detect NMD, we had also to introduce introns, and we have introduced 3 introns between exons 13, 14A, 14B, and 15, because, as I said before, without the exon junction complex, NMD is not observed. So we introduced in this mini gene the G542X mutation, the most frequent PTC mutation in cystic fibrosis, and we also added a flag tag at the 4th extracellular loop. So after RNA processing, this mini gene originates a messenger RNA, and in the case of the messenger RNA undergoing NMD, due to the presence of this PTC mutation, there is very low red fluorescence. And this is due to the truncated protein because it has very low levels, because the levels of messenger RNA are also very low. And there is no green fluorescence. In case of less NMD, so inhibition of NMD, we observe an increase in red fluorescence, but still no green fluorescence because the final part of the protein is not produced because it's a truncated protein. If there is some read-through, then the protein is produced in full length and we can observe some green fluorescence appearing. And in case the read-through introduces the wrong amino acid that actually prevents correct folding, we would not detect protein at the plasma membrane. So we have actually this flag tag at the 4th extracellular loop so that we can detect the protein which is present at the cell surface. So here I am showing the characterization of that novel construct, the mini gene, and the data included in this graph are quantitative RT-PCR from the wild-type mini gene. So this is the white bar on the left. And in comparison to the mutants, the G542X mutant mini gene, we can see that the levels of messenger RNA resulting from the mutant are highly reduced versus the wild-type. In the black bar, we have also the mutant, the G542X mutant mini gene expressed, but in the presence of the inhibitor of NMD, which is a chemical inhibitor. And we observe that the levels versus the MSO are actually very much increased, thus demonstrating that this mini gene undergoes NMD. Here are the same data actually showing in a Western blot the difference between, in lane 1, the mutant gene, so the protein resulting from the mutant mini gene, and in lane 2, the protein resulting from the wild-type mini-gene. And the reason why we cannot see any protein in lane 1 is because the antibody used here to detect CFTR protein actually recognizes a region of the CFTR protein which is absent in the truncated protein. And in contrast to the protein resulting from the wild-type construct, which actually has both forms, the processed form, which we call band C, the upper band, and also some of the immature form, the band B in the lower band. The other 3 lanes, 3, 4, and 5, are actually different constructs of the wild-type protein, either only with the GFP tag in lane 3 or at the C terminus, or just the M-cherry tag at the N terminus, or without any tag in lane 5. And M stands for the molecular mass markers. So we have tested these constructs under the microscope so as to detect the fluorescent labels. And here we have on the upper row the nuclei, which are not much different between the 3 types of samples analyzed. But we have on the left row the wild-type mini-gene with the 2 tags. On the middle row and on the right row, these are both M-cherry G542X EGFP construct. However, in the middle row, it's just the MSO, and in the right row, it's in the presence of inhibitor of NMD. So what we can observe for the wild-type gene, the one on the left column, is that there is no difference between the middle panel and the bottom panel because there is no NMD inhibition. However, for the G542X mini gene, we can observe for the bottom panel that there is a difference between the one just with DMSO and the one with inhibitor, because the inhibitor actually prevents NMD, so we see an increase in fluorescence. And here are the quantification of this data, so fluorescence for M-cherry, where we can see a significant difference for the G542X construct with the inhibitor on the far right, or just the MSO in the middle. So we use this construct, which we just characterized, in screens to identify genes that affect NMD and or splicing. Why splicing? Because, as I said in the beginning, the NMD process is actually coupled to the splicing because of the presence of the EJC complex. So to this end, we have used stable Hack flipping cells. Flipping means that our construct is actually inserted in a particular region of the Hack cell's genome. And we have screened the library of shRNAs, which actually targets 425 genes. So these genes are known to be involved in messenger RNA processing. Altogether, this was almost 2,000 shRNAs because each gene was targeted by 4 to 5 shRNAs. So we did the primary screen for NMD, which resulted in the identification of 13 genes actually inhibiting NMD, and 11 of these genes actually had more than 2 shRNAs, and 2 other genes just 1 shRNA. So here we also discarded some genes due to quality control. And then in the splicing primary screen, we actually identified 11 genes. So altogether, from the NMD and splicing screen, we identified 24 genes which actually resulted in some effect. And we submitted these 24 genes to a secondary screen for NMD, which identified 14 positive hits. And then we proceeded to hit validation with not SH, but siRNAs. under the same microscopy assay, also qRT-PCR and Western blot validation assays. So these are representative images of the NMD screen top hits. And here at the bottom, you can see the scores that we obtained for these genes. So basically, the last row is actually the one that was used to obtain these scores, and increase in the fluorescence versus the negative control, that is the scrambled shRNA on the very left, gives you the score that we have written here. So these are examples of the genes that we actually have pursued and resulted in the 11 top hits, which some of them also promote read-through, because you can see that inside the red boxes and in the middle row for GFP, we can see some increase in fluorescence versus the negative control on the very left. So here we are showing the effect of siRNAs, because before we had used shRNAs, in knocking down 6 of our best hits. So we can see that the targeted genes are actually the ones with the red bars at the bottom of the graph along the x-axis, and these messenger RNA levels are actually compared under conditions with the scrambled DSI SCR or the non-transfected control. And most of the siRNAs actually had an effect in reducing the levels of the targeted gene. So this is a proof that the effects of the siRNAs actually resulted from knocking down those genes. And we can conclude that in most cases, the knockdowns were actually effective. Here we have the effect of the same siRNAs on the expression of the CFTR messenger RNA bearing the G542X mutation. So what we can see is that at least one of the genes here represented by 43 with one of the siRNAs tested, siRNA number 2, actually had an effect in increasing the levels of the PTC CFTR messenger RNA. So we also wanted to validate by Western blot the effects of knocking down these genes, and what we observe here is truncated protein. So this is CFTR detected by antibody MM13/4, which actually recognizes the N-terminus of the protein. So even if the protein is truncated, this antibody will detect CFTR. Of course, the molecular mass that you observe here on the left is much lower than we usually observe for CFTR, because this is just truncated protein. And what we can observe here is that some of the genes, if not all of them, when knocked down, increase the levels of truncated protein versus the siRNA for scramble, the negative control, which is on the far right lane here on this image. Especially gene 43, which had an effect at the level of messenger RNA, is also confirmed by Western blot here to have an effect in increasing the levels of truncated protein, in particular with the second siRNA. So, in summary of what I've shown, a library of 425 genes related to RNA metabolism was screened by high-throughput microscopy. For NMD, 13 genes, or alternative splicing, which I did not show, but yielded 11 genes. And these 24 hits were rescreened and validated by qRT-PCR and Western blot using siRNAs. and at least one of the hits was consistent in both QRT-PCR and Western blot in increasing the RNA and protein levels, although truncated protein, resulting from the G542. I just want to stop here and actually acknowledge collaborators, acknowledge funding for this work, and also acknowledge my research group and the people who actually contributed to this work, and their names were listed when I presented the slides.
Hello everyone, my name is Jim Hu. I'm from the Hospital for Sick Children, University of Toronto. First of all, I'd like to thank organizers for the opportunity to share some of our work with the CF research community. Regarding the conflict of interest, I have nothing to declare. Cystic fibrosis is a multiple organ disease. For this audience, I don't need to give too much introduction, and except to say that currently the major morbidity and mortality is due to the lung disease. Fortunately, a major milestone has been achieved recently in developing drugs to treat cystic fibrosis. These drugs directly target the cause of disease, and they are called CF potentiators and CF correctors. So I actually classify the therapy with these drugs into three categories. Single drug therapy, so you can use ivacaftor, and double drug therapy, you can use lumacaftor plus ivacaftor or tezacaftor plus ivacaftor. And also triple drug therapy, you could combine three types of drugs. So these drugs can cover a large portion of CF patients. Now, we only have 5 to 10% of patients who require new therapies. It is nice to have these drugs. However, we don't know what the long-term effect of these drugs, whether these drugs can actually stop the lung functional decline of the CF patients. And we don't know the long-term side effects as well. Therefore, we need other types of therapy for the treatment. Of course, gene therapy is one of the alternatives for all these reasons. Airway is accessible to gene therapy vectors. Gene therapy can treat the cause of disease. Gene therapy can be designed to treat all types of CF mutations. And in patients with less severe mutations, if you have 10% of CFTR expression, and you can avoid lung complications. And also in vitro experiments, and people have shown that if you can get 10% of cells corrected, and then you can actually correct CF ion transport function. Early gene therapy attempts have shown that there are major challenges in conventional lung gene therapy. So these including we didn't have efficient vector system for therapeutic expression, and we didn't have safe and effective methods for vector delivery, and also there are immune barriers to vector delivery, and more importantly, it's very difficult to have therapeutic gene expression sustained in organs such as in lung, where normally you have cell turnover. Over the last 2 decades, many groups, including us, have taken all the challenges and made progress in many areas, such as in vector design and delivery methods. So in the next couple of slides, I will try to introduce some of the progress made in our lab. So this slide actually is showing the system what we're using. The vector system we call helper-dependent adenoviral vector. So I have to declare this is not our invention, and we just use the system. So I'd like to emphasize that the helper-dependent adenoviral system is not the same as adenoviral system, because in this vector we don't have any viral gene. So you can see that we only have a tiny bit of DNA sequences. And this is called terminal repeats, and the packaging signal actually are from the adenoviral virus. And so you can put actually 30 kb of the payload DNA. So most of the genes we don't even have such like 30 kb sequence. Therefore, you have to even put stuffer DNA in it. So when you produce this type of vectors, you need to have a helper virus. So in this case, this is just like the first generation adenoviral vector, and you have the packaging signal flanked by two LacZ sites. So when you have two types of DNA in the production cells, then this cell express a Cre and E1 regions. So the E1 will complement the first generation helper virus. So you can make all the proteins needed for viral replication and package. And so the Cre protein produced will cleave the packaging signal. Therefore, at the end, when you produce the vector, you only produce the helper-dependent adenoviral vector, so you don't package the helper virus. In 1997, our group developed a epithelium-specific gene expression cassette. Using this cassette and the helper-dependent adenoviral delivery system, our lab actually has shown that we can deliver genes into the airways of mice, pigs, and rabbits. In collaboration with others, we also have shown that we can get gene expression in the lungs of ferrets and baboons. As shown here, we can get relatively long-term LacZ reported gene expression in mouse lungs. As many of you know that if you use regular adenovirus, probably you can get expression for only a couple of weeks. In 2003, we have shown that we can protect the CF knockout mice from acute lung infection by delivering a helper-dependent adenoviral vector expressing the human CFTR gene. Although CF mice are relatively easy to handle and cost-effective, but they are not ideal for being used to test the CF functional correction. This is because there are major differences between airway immunology and airway biology between mice and humans. For example, there are major differences in terms of some mucosal glands, goblet cells, club cells, basal cells, as well as immune cells as well. That is why we tested airway gene delivery in different animals. Here the slide showing the LacZ report gene expression in the pig lung. 1 week following gene delivery, we took tissue blocks from different regions of the lung, and you can see we got extensive transgene expression. And here is a piece of lung from the right lung as a control. So in tissue sections, you can see transgene expression from the large airway, small airway, as well as alveolar region. We have also successfully demonstrated human CFTR expression in pig lung. The expression was shown by antibody staining, as seen here the green fluorescence. So this antibody is specific for human CFTR protein, not for pig CFTR protein. As you can see here, without gene delivery, you don't see any specific signal. So that means the antibody is specific. Because our vector does not integrate into the host genome, we cannot achieve permanent gene correction. One solution to overcome this is to integrate the CFTR gene into airway stem cells or progenitor cells. Fortunately, as shown here, in a human airway, there are a lot of stem cells or progenitor cells along the whole airway. For example, even in some mucosal glands, you have certain cells can be differentiated into other cells, basal cells, and you can see that they can actually differentiate into many different cell types. Even in alveolar region, the type 2 cells can be differentiated into type 1 cells. Airway basal cells are a good target for permanent airway gene correction. The question is, can you deliver genes to basal cells? And we have shown here that you can definitely deliver genes into the basal cells of mouse. We have also demonstrated that you can do it in pigs. For the safety of permanent gene correction, we need site-specific gene editing tools. Fortunately, recent advancements in engineering site-specific endonucleases, especially the CRISPR-Cas9 system, made it possible to achieve site-specific gene integration. The CRISPR-Cas9 system is a protein-RNA complex. It can be used to integrate a gene into a chromosomal location anywhere we want. When any new technology emerges, there are new challenges. For CRISPR-Cas9 system, here are the issues we have to deal with. So in the next couple of slides, I will try to address these issues one by one. When we talk about using CRISPR-Cas9 system for gene therapy, a lot of people worry about off-target effects. I think the whole issue is hyped because somatic mutations are well tolerated. Not every single insertion you could have a mutation or cause trouble. And also, Cas9 is site-specific compared to lentiviral vectors. And also the whole system can be further improved. So I don't think the off-target effect is a major issue we should deal with. On-target deletion could be a problem. However, if we use homology-directed repair, we use longer homology arm, that's also not an issue. Safe and effective delivery of the CRISPR system is an issue of concern because it faces the same challenges as conventional gene therapy. However, I've mentioned that some progress has been made in this area, including targeting stem cells. But Cas9 protein is immunogenic. This has to be dealt with. This slide shows our strategy to deal with the immunogenic problem of the Cas9 protein. We can package everything into a single vector, so when we deliver the vector into a targeted cell, the donor DNA part will be integrated into the location we want it, and the vector genome will be compromised. The rest of the genome will be degraded. Therefore, no more CRISPR-Cas9 expression anymore. Here we show that we can deliver both the Cas9 system and LacZ reporter into a single cell. And we actually used different vectors. This is just with different homology arm size. And the GFP is linked to the CRISPR-Cas9. So you can see that in all these cells we can get LacZ expression as well as the GFP expression indicate that we have CRISPR-Cas9 expression. Using junctional PCR with primers flank the homology arm, we demonstrate that we achieved site-specific gene integration. So you can see we can amplify a fragment, and this has to be confirmed by restriction digestion as well as by DNA sequencing. We use two different approaches to assess the gene integration efficiency. Here is the first approach. We transduce the cells, then we pass the cells for many generations to dilute the unintegrated vector genome. Then we see the cells, and then we stain the cells with xGAL, and then we count the ratios of blue and white cells. And here is the efficiency. It depends on what type of vectors you get different efficiency. Roughly, we can achieve around 10%. Here is our second approach. We first transduce the cells, and then we sort the cells each into a single well of a 96-well dish, and then we stain the colony. And you can see if you have a gene integration event occurred, the whole colony will be blue. So in this case, we estimated pretty much similar efficiency. And if you use a chemical called SGR7, you can enhance the gene integration efficiency a little bit. Most importantly, we found the Cas9 expression diminishing very quickly after transduction. This has been confirmed by both real-time PCR at the messenger RNA level and Western at the protein level. Consistent with the observation of disappearance of Cas9 expression, we found that the vector genome in transduced cells diminishing very quickly as well. And we have confirmed this in two different cell lines. And as you can see that in single cell colony, we don't have any vector genome anymore. And so this is the control. In addition to our work in human cells, we created a CFTR pig knockout cell, and we actually confirmed our results in pig cells. And we don't have time to show you everything, except I just show one slide. So this is actually a flip assay to measure the CFTR activity of the cells following vector transduction. And you can see here in passage 0, no matter you have a CRISPR integrated vector or you have a regular vector, you can see CFTR activity. And after the cells pass for 10 or 20 passages, now you don't see CFTR activity in these cells. So this is the flip assays is measure the membrane potential. You can see when you add forskolin, you see CFTR activity, and after you add CFTR inhibitor, you can see the activity can be inhibited. And so this is just to summarize. So for example, like in passage 10, and if you have the negative control or you have a regular vector with no gene integration, you don't see CFTR activity. And here you see nice CFTR activity. So 20 passages is pretty much the same. Now, the last issue is gene targeting efficiency. So we know that actually in vivo it's not very easy to achieve high gene integration efficiency. Fortunately, there are many different ways we could enhance efficiency. For example, use small molecules or use proteins involved in DNA repair or cell cycle regulation, or even target more than one loci. And even more, we could explore the non-homologous end-joining pathway. In summary, we made progress in gene delivery to airway epithelial cells, including basal cells, We also made progress in the CRISPR and donor DNA. And also, there are ways to improve the gene integration efficiency. Finally, here is a summary of our gene correction strategy. We hope to use a single viral vector to deliver CRISPR and donor DNA into the airway stem cells, and when the cells receive the vector, we'll be able to integrate the genes into whatever locus we wanted. Finally, I have to thank the people who work with me and who collaborated with me. Thank you for your attention.
My name is Marcus Mahler. I'm the director of the Department of Pediatric Pulmonology, Immunology, and Critical Care Medicine in the Cystic Fibrosis Center at Charité University Medical Center in Berlin. And I welcome you to this on-demand symposium where I will speak about mucus biology and implications for CF therapy. Here are my disclosures, including personal fees for consultation, non-personal fees for participation in clinical trials, as well as research grants related to the development of new therapies for cystic fibrosis. And here's a key problem in CF that is therefore also known as Mucoviszidosis, and that is increased viscoelasticity of the CF mucus. That results in impaired mucociliary clearance and mucus plugging, which in turn causes impaired ventilation, forms a nidus for chronic bacterial infection that then triggers chronic airway inflammation, which plays a key role in the progressive structural lung damage of our patients. Mucus is essential for health, as it is an important component of normal the mucociliary clearance system, as shown here, and it's been complicated to study because the two main molecules forming this mucus gel, MUC5AC and MUC5B, as you can see here, are large and complicated in the structure, complicated glycoproteins that consist of von Willebrand factor-like domains at the NNC termini that are cysteine-rich, where the monomers dimerize via disulfide bonds. The core of the protein is rich in threonine and serine, where it's heavily glycosylated, providing water-drawing properties as well as negative charges. And then there are other cysteine-rich domains that can contribute to intra and intramolecular disulfide bonds that contribute to the complex structure of the polymeric mucus network. In cystic fibrosis, this system fails and mucus accumulates in the airways, and this failure has long been related to increased viscoelasticity that can classically be measured by mechanical rheology, as shown here, showing an increased elastic as well as viscous modulus of CF compared to healthy mucus. So the question, what determines this increased viscoelasticity of the CF mucus gel, has really been a holy grail in the field. And several determinants have received attention in this area, also with respect to their relation to CFTR dysfunction. And these are hydration, the role of cross-linking, and the role of bicarbonate, calcium, and pH. And in my talk, I now want to review the current evidence for these determinants and their role, as well as potential therapeutic implications. First, hydration. It's long been known that deficient CFTR chloride and fluid secretion results in airway surface liquid depletion or airway surface dehydration. And what's been shown mainly by the UNC group over the past decade is that this airway surface dehydration also results in a dehydration or hyperconcentration of the mucus gel layer, and that this then also results in an increased osmotic pressure, and that this increased osmotic pressure of the gel layer from a point where it exceeds the osmotic pressure of the paracillary layer can then cause osmotic compression of the system that's above a critical threshold of about 6% solids and 94% water, then causes a progressive slowing and impairment of mucociliary transport. Regarding therapeutic interventions to improve mucus hydration, we for quite some time have inhaled hypertonic saline, and from clinical studies that have been performed, we know that acute treatment with inhaled hypertonic saline indeed improves mucociliary clearance and lung function in adults. And more recently, in the SHIP and PRECIS studies, it was reported that long-term inhalation in those studies over a period of 1 year improves the lung clearance index as an outcome measure for small airways mucus obstruction, both in preschool children and infants, supporting that improving hydration has therapeutic benefits. Second, cross-linking. The issue is that CF mucus has polymers and abnormally formed structures that are not responsive to hydration. And these stiffened or permanent gels found in this study by John Fahy's group contribute to the increased viscoelasticity. They contain an increased concentration of cysteines and disulfide bonds, and these disulfide bonds correlate with reactive oxygen species, leading to the concept that oxidation of the cysteines leading to excess disulfide bridges leads to increased cross-linking. That is another key problem in CF mucus. In this study, they also developed and tested new thiol-modified carbohydrates, as shown here. And what they found is that these molecules are more effective in reducing viscoelasticity than the gold standard molecule N-acetylcystein. And recently we had an opportunity to study one of these new reducing agents in the beta-ENaC transgenic mouse, where in a preventive setting, i.e. starting the treatment from birth for a period of 2 weeks, we could show that these thiol carbohydrates reduced the spontaneous mortality as well as the intraluminal mucus content in this mouse model. And additional information on these studies will be provided by Annalisa Ardante on her poster and workshop session. Very similar data were reported from another new reducing agent, P3001, that also was superior compared to N-acetylcystein in reducing sputum rheology and improving mucus transport rates on primary bronchial epithelial cells, as well as reducing mucus plugging in the beta-ENaC transgenic mouse. So taken together, these results really suggest that these new reducing agents are promising to reduce mucus plugging and have beneficial effects in vivo. Third, bicarbonate, calcium, and pH. In addition to chloride, CFTR also conducts bicarbonate, and therefore the role of bicarbonate has been of interest for a long time. And in 2008, Paul Quinton published a hypothesis that predicted that this bicarbonate secreted by CFTR onto the airway surface is important for the sequestration of calcium from negatively negative charges in the mucins that are packed with high concentrations of calcium in the granule, and then upon release, this sequestration will expose negative charges, lead to electrostatic repulsion and rapid expansion, whereas in the absence of bicarbonate, this divalent bridging between calcium and the negative mucin charges persists, impairing proper expansion and transportability. This hypothesis was first tested in the CF mouse ileum, where it was found by Gunnar Hansen's group that adding increasing concentrations of bicarbonate makes the otherwise sticky CF mucus more mobile in a way that it could now be removed by suction from the epithelial surface, and very similar results were obtained when the free calcium was reduced by adding EDTA. More recently, this concept was also further supported by electron microscopy studies on native isolated MUC5B polymers, as you can see here. And the finding was that in the presence of high calcium, low pH, the MUC5B polymers acquire a more condensed, compacted configuration, whereas under conditions with low calcium or neutral pH, they are more expanded and linearized, again supporting the concept that high calcium and acidic pH lead to a more compacted and cross-linked MUC5B, whereas under neutral pH conditions and low calcium, the molecule is more linearized and expanded. Regarding therapeutic intervention, there's a recent study from Steve Rowe's group looking at the combination of bicarbonate, glutathione, and ascorbic acid, and showing by optical coherence tomography on primary bronchial epithelial cultures from CF patients, as well as using particle tracking microreology, that this combination improves mucociliary transport, makes particles in the mucus more mobile, and reduces viscosity of CF mucus. Interestingly, when they try to dissect which of the components determines the therapeutic effect, they found that bicarbonate plus glutathione was most effective, suggesting that in addition to bicarbonate, the antioxidative properties of glutathione may have contributed to this beneficial effect. Last but not least, CFTR modulation. Obviously, with the emergence of effective CFTR modulator, I think it's a key and fascinating question to ask to what extent they can normalize CF mucus. The data we have so far, first are MCC data in patients with a G551D mutation that started ivacaftor therapy. And as you can see here, ivacaftor improved mucociliary clearance after 1 month, that persisted for 3 months, both in the central airways as well as peripheral region of the lung. Second, I think really fascinating study from John Engelhardt's group in the G551D CF ferret, showing that very early start of ivacaftor treatment, in this case means in utero, can actually prevent the onset of mucus plugging both in the conducting airways as well as submucosal glands completely, whereas withdrawal of ivacaftor after birth then again results in the formation of these CF-like mucus plugs, which I think is really supporting and fascinating data on the histopathology level. And in the next study, in a now CF rat with a humanized G551V CFTR mutation, by again using optical coherence tomography and particle tracking microbiology, it was found that restoring CFTR function by ivacaftor indeed has beneficial effects on ASL and PCL depth, as well as mucociliary transport in across the surface of the native rat trachea, in that it improved particle mobility and movement and resulted in a significantly improved viscoelasticity of this CF rat mucus, which I think is really promising that restoring CFTR function has beneficial effects on the CF mucus abnormalities. Briefly, want to summarize my presentation. Mucus dysfunction plays a central role in the pathogenesis of CF multi-organ disease. Studies of mucin in mucus biology have identified hydration, cross-linking, bicarbonate, calcium and pH as important parameters of mucus structure and function, and have provided insights into the mechanisms of mucus dysfunction and cystic fibrosis. Novel therapeutic strategies based on these insights are at the moment actively tested in the preclinical arena and have the potential to improve mucus function in CF and potentially also other diseases. And emerging effective CFTR modulator therapies obviously provide an unprecedented opportunity to correct the CF mucus defect, but the level of correction in patients, also in the context of their stage and severity of disease, obviously remains to be determined, and I think this will be a key research question for the near future. With this, I'd like to thank you again for joining this on-demand symposium, and I'd be very happy to take any questions you may have either in the chat or the conference platform. Thank you.
Greetings. My name is Rabindra Tiruvandiam. I'm a PhD scientist at Emory University and Children's Healthcare of Atlanta. And it's my pleasure to stream this presentation from my office here at Emory on inflammation and opportunities for therapy for all with CF. So I want to thank you first for tuning in, and also the chairs of the session, Dr. Jonker and Quentin, for the invitation to speak. I have no relationship to disclose. This presentation will be organized around 3 parts. In the first one, we're going to look over the data on inflammation in CF, topology, mechanisms, clinical impact, and based on this, look at the current opportunities for therapy, what to target, why, and how. And finally, we're going to try to put everything together in perspective and update the playbook on inflammation in CF so that we can pave the way for future research and interventions. So let's start with the introduction on inflammation in CF. Inflammation is defined as a response to severe stress or injury that's marked by leukocytic infiltration into a tissue, and that initiates the elimination of the stress, which could be sterile damage to the tissue or damage due to agents like bacteria or viruses. In CF, the stress is really coming from dysfunction of the CFTR, which leads to problems with the function of exocrine organs. By and large, even though CFTR is expressed throughout the body, the inflammatory manifestations of the disease are primarily in the lung, in the GI tract, and in the blood. And we're going to be talking primarily the lung today, because this is where most of the action is. So the lung is really composed of three parts: the large airways, the small airways, and the alveoli. And in CF, early on, right after birth and during the first few years of life, the action is really in the small airways. Epithelium that's lining the lung surface, and also macrophages that are resident, are somewhat dysfunctional, and they allow for the recruitment of neutrophils that come from blood. Later on, in late childhood and early adulthood, you have what we call chronic CF inflammatory disease, which is sort of a metastable state that sometimes gives way to those acute pulmonary exacerbations where disease flares. And this is happening primarily in the small airways, in the large airways, and to a lesser extent in the alveoli. Same cells are being involved. This devolves into the destruction of the lung, really, such that patients with CF might become eligible for a transplant. After transplant, you have inflammation again occurring in the lungs, small and large airways, and this time, in addition to the three cell types we talked about already, T cells are also involved. Obviously, the pathology pre-transplant and post-transplant are very different. So we're going to be talking mostly about what's happening in early and chronic CF pre-transplant today. If you look at a slide, of a CF tissue, what you see that's really striking is this accumulation of neutrophils in the lumen of bronchi. It's early, massive, and sustained. And this is to paraphrase a paper from Dr. Quentin. What's happening also with these neutrophils is that they don't just hang around the lumen, they release the content of their granules. And some of these molecules are very, very toxic. So neutrophil elastase is a major one. It is a protease, so it leads to proteolysis of the extracellular tissue. And it also leads to induction of mucus production by the epithelial cells. In the same granule as NE, myeloperoxidase, which is the enzyme that makes bleach, is released also, and that leads to the formation of a lot of oxidants in the lumen. This occurs regardless of mutations of the CFTR. So you can have mild mutations of the CFTR and have neutrophilic inflammation, okay? And this is also very paradoxical because neutrophils are made to take care of bacteria, but in the context of CF, this massive inflammation with neutrophils coincides with the inability to clear infections. So that has been really vexing for researchers for a long time. Until I think our group really paid a lot of attention to this, and we went directly into patients' sputum and conducted deep phenotyping of the neutrophils that we collected from that really nasty environment. And what we found is that the immunology playbook needed to be updated. In fact, the CF lung was showing the presence of a fate of neutrophils that hadn't been described before, which was not the typical fate where bacteria are taken in and killed in the phagosome, the phagocytic fate, or one where neutrophils release their DNA explosively to make extracellular scaffolds that can trap bacteria. This is the netotic fate. But we found a fate where the primary granules that contain elastase and myeloperoxidase are actively exocytosed. So they're put to the surface and they open up and the elastase and myeloperoxidase are released into the lumen. So these cells are granule releasing, but because these granules also contain arginase 1, which is a very strong inhibitor of macrophages and T cells, they're also immunomodulatory. And because these cells keep living for a while, they're very active. They activate the mammalian target of rapamycin pathway. They're anabolic. They take up a lot of glucose and inorganic phosphate and amino acids. They're metabolically active as well. So we call them GRIM. Okay, and it's also not a good omen to have them in the lumen of the lung. What we found is that these GRIM neutrophils are present as early as 3 months of age and throughout life in patients, and they're the major source of neutrophil elastase and myeloperoxidase in the fluid, and they do that by active exocytosis. What's interesting is that we can reproduce this phenotype in vitro by growing air liquid interface cultures of small airway cells and exposing the apical surface to either a chemoattractant or to supernatant from CF sputum that has been rid of bacteria and cells. And we can migrate naive neutrophils from blood through this layer. And in the context of the CFASN, the neutrophils mimic exactly what we see in vivo. That is, here if you compare blood to sputum or pre-transmigration to post-transmigration into the CFASN, there's an increase in CD63, which is a marker of the exocytosis of those granules, and a decrease in CD16. CD16 is cleaved by elastase. And concomitant with the formation of this green phenotype, we could see very clearly, both in vivo and in vitro, that green neutrophils accumulate a lot of RNA. So what we did was we actually inhibited de novo transcription by alpha-amanitin. And what we found is that in the context of CFASN, this leads to a decrease in the release of elastase, but also this leads to an increase in the killing of bacteria. So this is very exciting because now we have the indication that the green phase is induced by exposure to the CF airway fluid, and it doesn't really matter whether you're a healthy control or CF blood neutrophil, you're gonna react the same way after transmigration. And the green neutrophils not only exocytose their granules, but they actively repress bacterial killing through de novo transcription. So now that we've got that established, how is it really impacting, the health of the patients? So what we know now after years of clinical studies is that the burden of elastase and myeloperoxidase in early CF, chronic CF, APEs, and even post-transplant is linked to pathology. But what's really interesting, and this is something we've done in the last decade, is we found both in early CF and in chronic CF that the burden of green neutrophils this time, the cells that actively release elastase and myeloperoxidase, in early CF is linked positively to structural lung damage that we measure by CT scans in babies, and also in older children and adults. The burden of green neutrophils in the sputum correlates inversely with lung function that we measure by spirometry. So what are the opportunities for therapy? Can we target these cells, and can we target other cells? Why and how can we do that? So the why. What are the goals of therapeutic interventions aiming at CF inflammation? In early CF, obviously what we want to do is we want to delay the start of the structural lung damage, and we also want to stem its progression once it has started. We want to prevent the colonization by bacterial and fungal pathogens. Once disease has progressed in older children and adults, in the small airways and the large airways, what we want to do is we want to disrupt the casts that are made of neutrophils that are surrounding bacteria but not killing them. We want to restore killing. We want to reduce the severity of the acute episodes, and we want to promote full recovery after them. And over the long run, this is going to preserve lung function. In the context of transplant, we want to also control inflammation so we can prevent rejection and prevent infection of the grafts. So what are the opportunities for therapy when we look at those green neutrophils? First of all, everything we talked about, that they hyperexocytose, they fail to kill bacteria, they release molecules that are immunoregulatory or toxic, and they undergo transcriptional adaptation. Well, we can target that with specific inhibitors. But we can also, with regards to transcriptional adaptation, try to modulate it with either small molecules or with gene therapy. With regards to the netosis fate, this is a short-lived fate of the neutrophils where they release those neutrophil extracellular traps made of DNA, and they also release their granules in that context, where we can use the same inhibitors of those granule contents, but we can also look for specific inhibitors of the DNA release. So I'm going to illustrate a little bit here what we did recently with regards to exocytosis inhibitors and maybe proof of concept for transcriptional modulation of the greens. This is part of an ongoing study that's funded by the CF Foundation, where we're screening for new molecules that are going to be able to promote killing by the green neutrophils. And we can find here one of the drugs that we found, drug X. I cannot disclose it so far, but suffice it to say that in our system, this drug can basically promote decreased exocytosis of the primary granules that contain elastase and myeloperoxidase by the green neutrophils. but they can also, that drug can also promote the killing of bacteria to the level of the sort of control, healthy lung neutrophils. How can we target these cells in vivo though? Because they're in the middle of the airways. Can we find a way to bring a cargo, either a drug or a gene, directly to them? And this is something we did with Amit Gagar at UAB and Chris Roy at Georgia Tech. The idea here is to weaponize the fact that neutrophils, the GRIMs, release elastase. So we can engineer those microgels that basically have ropes around them that are elastase sensitive. So when they're sent to the lung, they swell, they reveal the ropes. Wherever elastase is enriched, they're going to undo themselves and release their cargo. If the cargo is a nanoparticle that contains an inhibitor of exocytosis, then we can actually block the release of elastase. And we can also block both blood and lung inflammation in a single dose. This has been published recently. Here I'm showing another application of this technology. Instead of putting a drug in the nanoparticle that's in the microgel, we put RNA. And we can find in that case that we easily express in the green neutrophils whatever RNA we want. So that can be a small interfering RNA, that can be a therapeutic RNA. So this is really paving the way for gene therapy of these cells. Beyond those small molecules and beyond gene transfer to the neutrophils, can we target other cells? Obviously, neutrophils, I put them here in the middle of the picture, but they are interacting with T cells, B cells, NK cells, monocytes, macrophages when it comes to other leukocytes, the epithelium, and also the microbiome. So all of these components are adapting to the chronic presence of green neutrophils, okay? So can we do anything? And instead of targeting the neutrophils, target the other cells. So I'm just gonna go quickly over some of the potential options we have. One is, how can we allow the macrophages to take control back from the neutrophils? How can we increase their poise? And the idea here is really to target two pathways that we found to be very active in CF that are decreasing the ability of macrophages to do their job. One is the downstream of the release of arginase by neutrophils. Arginine is essential for macrophages to do their function, especially to activate NO production. And downstream of arginase activity basically is a lot of, of activity from macrophages. So what we want here is to inhibit this pathway so we can make the macrophages active again, especially in bacterial killing. We found also another way by which macrophages get inhibited by neutrophils is through the induction of PD-1 signaling, which is an exhaustion marker. And this is a pathway that's been used therapeutically in cancer with a number of different anti-PD-1 antibodies. So this is something that could be envisioned potentially also in CF. T-cell poise is also dependent upon the ability of arginine. So arginase activity that's released by neutrophils by exocytosis, this can also be reactivated by inhibition. So we think arginase inhibition can actually have a lot of interesting sort of positive results with regards to inflammation in CF. We can also look for the epithelium. The epithelium is the original agent in promoting the inflammation in the lung. And this is something that we're actively pursuing as well as a group. And we think the same type of small molecule treatment or gene therapy can target the specific transcriptional adaptations going in the CF airway epithelium that's responsible for inflammation. Okay, so how can we take all of this knowledge and update the playbook on inflammation in CF? Well, first of all, we need to look at other pathways. It's not just about cutting the faucet and preventing the recruitment of cells into the lung, which we've done for decades. Because if the cells are recruited, first of all, is because there's a lot of redundant ways to recruit them into the lung. So no single way to cut the faucet or the flow will work. But it's not just that. If they're recruited, it's for a reason, right? So what we want is really to go at the root of what's causing the neutrophils to come in the first place. So we still don't know what it is actually. Downstream of CFTR mutation, something is wrong in the epithelium, and the properties of the airway fluid are changed. And this is why when we take this altered fluid, put that in vitro, we can green the neutrophils in our transmigration chamber. Is that coming from an intrinsic defect, or is that coming from maybe reflux from the GI tract? Can we right those wrongs in terms of metabolic imbalance, dangerous signals, potentially GI reflux, increased burden of mucus? Can we also, downstream of all of this, instead of focusing on neutrophils, look for ways to, as I said earlier, promote better poise for the T cells and macrophages that are supposed to provide some control over neutrophils. So these are examples of the new pathways I think we can target. So we can expand studies to those new pathways. We should also expand our studies of inflammation in CF to other subsets. I highlighted epithelium, macrophages, neutrophils. We talked a lot about them today. There's a lot of other cells that are resident in the lung or that are recruited from blood into the lung. And we don't really know what these cells do at the different stages of CF disease. So that's really something that I look forward to in the future. And finally, I think if we want to look at CF disease, we need to be able to sensitively measure it in a way that's less invasive. I showed you some data on BAL, which requires sedation and bronchoscopy. We do a lot of our surveys of the structure by CT scanning. So can we replace CT with lung clearance index, for example? Can we also replace the collection of bronchoalveolar lavage with induced sputum, which is less invasive, or even better with exhaled breath condensate? Are some of the biomarkers of tissue inflammation showing up in blood? And can we get those also through whole blood? So this is part of a study, a workflow that's part of a study that's also funded by the CF Foundation in our institution here at Emory, where we actually do this in babies with CF from 6 months of age and on. And then we need also to target emerging issues that have high relevance to inflammation. I'm going to go quickly over those. You know about them. The impact of the modulators. Are they changing the immune system in CF? Are they improving inflammation? Can gene therapy of the CFTR do that as well? But also, how is it going to impact inflammation? Because inflammatory cells have scavenging function that can interact with the vectors of gene therapy. And trained immunity to the vector or transgene product can also be a problem. Finally, how is inflammation in CF interacting with new pathogens? The impact of chronic infections by CMV, EBV, but also SARS-CoV-2. Obviously, we're all very interested in figuring this out. I'm going to stop here and acknowledge my laboratory, also my collaborators in the US, international collaborations, funding by CF Foundation, DARPA, NHMRC, NIH, and NSF. And I'm going to finish also here in the bottom by providing my email and my webpage so that you all can contact me if needed. And again, thank you for your attention.